is Felix, and my supervisor is Professor Mukerabigwi. Here is the outline about my topic, and then please allow me to start with the first part of my presentation, which is the research background of my research. It is known that cancer is a growing burden to mankind and ranked the second among serious diseases, leading to major death worldwide. For example, in the year 2018, around 18.1 million people of new cancer incidents have been recorded. And also it is estimated that nearly 10 million people died because of cancer. So when we go to see about the treatment, chemotherapy is the main systematic treatment option for a wide range of cancers. However, it is less effective, mostly due to the high clearance during the blood circulation, severe side effects, as well as multi-drug resistance. When we come back on the multi-drug resistance in cancer, multi-drug is often encountered during the treatment. However, and also represent a real obstacle for the effective treatment. And about the major cause about multi-drug resistance, here we can list, for example, membrane drug transporter, cell damage, self-repair, such as DNA self-repairing during the treatment, and also alteration drug target structure, as well as activation of various detoxification mechanisms in the cell. So to solve the previous question, since there are several decades, there have been great interest into the application of nanotechnology in cancer treatment. For example, nanoparticle can be used as the vehicle to deliver various drugs within the cell. Then, for example, within the help of the various stimuli, such as intrinsic stimuli or extrinsic stimuli, the drug can be released within the cell at the tumor site, which can improve the treatment efficacy. Compared to the small molecule, nanoparticle present various advantages, such as one can achieve increased systemic circulation, half-life, as well as which can also improve the drug viability at the tumor site, and also it can achieve reduced adverse effect. So among the nanoparticles, recently there has been a great interest about the application of therapeutic nanoreactors in cancer treatment. So briefly, nanoreactor can be defined as nanocompartments which are characterized with the confined reaction space containing immobilized catalysts and able to produce functional product locally. Here from this picture, we can see the key characteristic of therapeutic nanoreactor. It should have structural compartment, and also it should have the selective membrane which can allow the diffusion of several molecules, for example, such according to the molecular weight or various options. And also it can be used to achieve targeted delivery, as well as can be used to encapsulate enzyme. And also when it finishes its job at the treatment site, it should also self-destruction. Compared to the conventional nanoparticles, this therapeutic nanoreactor have their unique advantage. For example, it is feasible to immobilize the active molecule without harming their activities, such as enzyme. And also, the reaction vessel, as we said before, the membrane can be selective, which can allow some molecule to cross across the membrane while retaining the other molecule, for example, inside. However, among the existing challenges about nanoreactor, it is still complex to prepare those therapeutic nanoreactors. Again, most of the previously reported nanoreactors have the intrinsic permeability, which can cause the unwanted side effect. And also, the requirement of multiple steps for cancer treatment. For example, in EPT, which is enzyme product therapy, where there will be requirement to deliver like activating enzyme first, and also the product second, which can affect the therapeutic effect. So here I come on the main objective of my dissertation. The first one was to develop a facile approach to engineer therapeutic nanoreactor with branch key features, which are, for example, the tunable membrane permeability, which is more important, and also the selective transportation. And also to develop a nanoreactor which which can be only activated at the tumor site. Another objective of our research was to develop various treatment strategy to reverse multi-drug resistance during cancer treatment, for example, through the amplification of anti-tumor oxidative stress, as well as targeting mitochondrial organelle metabolic activities. Here, in the second part, we are going to talk about the engineering of tumor-specific nanoreactor to achieve single-step enzyme product therapy, which has been abbreviated as EPT. Actually, the enzyme product therapy is a treatment strategy which has been designed to improve the treatment efficacy and also by reducing side effects. For example, a non-toxic product can be delivered, but when treated at the interested treatment site, and then the non-toxic product can be activating toxic drug by different mechanisms, such as in the presence of the end user. However, this strategy still requires separate delivery of activating enzyme as well as a product in most cases of EPT, which has been previously reported, which can also cause the less variability and possible activation of that, which can cause unwanted cytotoxicity, which also will impair the compromised therapeutic effect. So here, we design a therapeutic nanoreactor which can be used to achieve a single-step enzyme product therapy, which means it can deliver both product and enzyme at the same time. And also it can achieve on-demand specific nanoreactor activation, which means it can be activated only at the tumor site. Here, this is how we did it. First, we prepared a polymer, which has been self-assembled into polymer zone and used to deliver both activating enzyme, which was glucose oxidase, and the product has been synthesized based on the common anti-cancer drug, which is CPT, and then we name it for CPT. And then it has been done such as during the systemic circulation, it can remain inactive, but when treated at the tumor site where there is weird acidic condition, it can release high amount of H2O2, hydrogen peroxide, to induce high amount of the ROS, the active oxygen species in the cell, and also it can release the toxic. After this product, it should be not less or not toxic to normal cell. But this, when it is activated in the tumor cell, it is toxic to the cell. Such as this combination can achieve improved therapeutic efficacy. Our polymer has been synthesized on the last polymerization using this two monomer, which is benzene methacrylic, and then another monomer which is contained tertiary amide, which is ultra pH sensitive, such as our polymer is pH sensitive. And then our polymer has been fully characterized with enema as well as the DPC short symmetric cap, which means this polymer can be used to sample into uniform morphology. So we use our polymer to self-sample into polymer zone. As we have determined that the pK of our polymer is of our polymer zone 6.9. It means it can respond to the pH when it is incubating the pH, which is lower than its pK. However, we found that our polymer nanoreactor, which has been abbreviated like this, which was loading the glucose oxidase enzyme and our product, which is porosity, short structure, stability, both pH, which is pH 7.4 and then pH 6.8. And here we get the membrane pH sensitivity of our nanoreactor using a colorimetric reaction. Here, this is how we encapsulated the glucose oxidase, our polymer zone, and then we incubated into the medium condition at pH 6.8. And then outside the polymer zone, we put glucose in the presence of HRP enzyme and also in the presence of colorless chemicals, such as when this pH can impede the membrane permeability to become there will be a fast transition, such as small molecule can enter inside the polymer zone and also initiate the reaction which can produce high amount of H2O2. This H2O2 can be used by HRP to oxidize chemicals such as the the blue color can be observed. According to this result, it was observed that our nanoreactor membrane was selective permeable at pH 6.8, but it was not found that it was permeable at 7.4, which means our nanoreactor can be used for further experiments such as for cancer treatment. Here also we studied the selective and tunable membrane permeability of our nanoreactor. We synthesized a series of polymer, and then those polymer, only they were different in the pH responsive segment, starting from 0 up to 30%. And then it has been found that the polymers which can be synthesized by using this ratio of pH responsive, it can retain high molecular weight material inside, but it can release low molecular weight material, which means it can retain high molecules such as inside, but release small molecules. Here, this is how we synthesize our product. We started from common and cancer agent, which is camptothecin, CBT, and then by using very chronic ester, we have fetched our drug, such as this drug cannot be toxic when we don't want to be toxic. But in the presence of hydrogen peroxide, this hydrogen peroxide can trigger self-immolation from this side, this boron side, and convert this precipitate back into the parent toxic drug, which is CBT, which means if we activate this organ in the presence of H2, we can come back to the toxic drug. And also our product has been fully characterized by NMR and mass spectroscopy as well as the HPLC before and after the product. Next, we have, because our nanoreactor can produce hydrogen peroxide, and also it should release the CPT. We studied the hydrogen peroxide release at different conditions, and also we studied the CPT release at different conditions. In short, it was found that our nanoreactor can effectively generate and release both hydrogen peroxide as well as release the toxic drug at pH 6.8, but at pH 7.4, there was negligible release of the drug as well as negligible release of the H2O2. Next, we moved into the evaluation of intracellular ROS. According to this figure, when our nanoparticle or our nanoreactor is incubated at the tumor pH, it can allow small molecules such as glucose and oxygen to enter inside, and then which can stimulate the production high amount of H2O2 and also reduce the CPT. Here, about the intracellular ROS generation, it was found that our nanoreactor can obviously induce the generation of intracellular ROS at pH 6.8, much more compared to other treatments which have been Next, we have studied the in vitro cytotoxicity and as well as live and dead assay to see how cytotoxic can be our nanoreactor. And it was found that our nanoreactor was toxic. We used the A549 cell line, but we did the condition at different conditions. We did the experiment at different conditions, which was at pH 6.8 as well as at pH 7.4. It was found that our nanoreactor was much toxic. This blue color was much toxic to this cell line at pH 6.8, but when it comes to pH 7.4, there was less toxicity, which means that our nanoreactor was cytotoxic at pH 6.8 with negligible cytotoxicity at pH 7.4. This means that if our nanoreactor can be used, it can only kill the tumor cell, but also sparing the normal cell, which means there should be no undesirable cytotoxicity. So this result has been also confirmed by live and dead, where much of many cells were PI positive. The red color means the dead cell, the green means the live cell. We also tested our nanoreactor in vivo, and then first we started the in vivo bioavailability. As can be seen here, the tumor site, even after 48 hours, there's still appreciable amount of our nanoreactor. but still also it can also be observed as some nanoreactor in the river. However, we also tested the intratumor ROS, which it was external that our nanoreactor can produce high amount of ROS intratumorally. Then it was found that our nanoreactor can be accumulated at tumor site, and also it can be used to induce high amount of intratumor ROS. Next, also we tested the antitumor efficacy of a nanoreactor. It was found that our nanoreactor of interest can remarkably inhibit the tumor growth compared to other treatment groups, and also it can severely damage the tumor tissues, according to this histological analysis by this H&E image at the tumor tissue after the treatment. So in summary, we presented a nanoreactor that can encapsulate activating enzyme as well as prodrug, which can be used, for example, to achieve a single-step enzyme prodrug therapy. And also, it showed tumor-specific as well as specific membrane permeability. Moreover, the approach used to produce or to synthesize our prodrug can be by caging with phenylbromide ester cage, can be fastly extended to many other anti-cancer agents, especially those containing amide or hydroxy functional groups in their structure. Please allow me to move to the part 3 of my talk, which will be about the application of nanoreactor to reverse multi-drug resistance. Here, we come on the cisplatin, which is a common anti-cancer agent, which is frequently used in the clinics. Normally, if the cells are not resistant, this is the mechanism which cisplatin can induce the cytotoxicity. When cisplatin is enter into the cell, it will produce DNA adapt. This DNA adapt, which means it will induce, it will inhibit DNA self-replication, and then there will be production of several damage recognition proteins, which will, for example, produce caspase, and then which will induce the apoptosis, and then the cell will die. However, this drug is often encountered with drug resistance during the treatment. If there will be drug resistance against this protein, this proposed mechanism can take place. When drug enters the cell, the drug can be found out in the cell by various Felix membrane transporters, and also which will reduce the DNA damage, and also this damage recognition protein will be reduced, as well as there will be activation of cell cycle RS transcription factors, such as those in blue color. And then there will be reduced amount of caspase, such as caspase 3, and then the apoptosis will be severely reduced, which can enhance cell survival, even if there will be the presence of cisplatin during the treatment. So to try to solve those problems, we asked ourselves a question: can amplification of intracellular oxidative stress sensitize cisplatin against resistant cancer cells? And here we come with synthesis of a nanoreactor, which was quite similar with the previous part. And then we use this nanoreactor to encapsulate both enzyme, which is glucose enzyme, glucose oxidase. This glucose oxidase is also one of the exogenous therapeutic enzyme which can induce the cytotoxicity to the cell, especially by enhancing the production of ROS and also by consuming the cellular nutrients such as glucose. And also, this nanoreactor at the tumor site, it can release this ROS, which is hydrogen peroxide, and then there will be release of cisplatin. This release of ROS can induce the apoptic pathway such as caspase 3. Then the release cisplatin also can inhibit the DNA replication, which in combination can achieve cell death. So we move, this is how we prepared our nanoreactor. We prepared this polymer, which has been self-assembled into polymerism to encapsulate both GOT and cisplatin. And also, our nanoreactor has been used for several experiments such as cell cytotoxicity. Also here, because we are dealing with resistant cell, we use two different, the same one cell line, but those cells were different. They are resistant cell and non-resistant cell at both pH 6.8 or pH 6.8. And also we performed the same experiment, but at pH 7.4 and pH 7.4. And according to this result, it was found that our nanoreactor can not only achieve the cytotoxicity to the non-resistant cell, but also resistant cell, which was comparable. And then on the other side, when the treatment has been conducted at pH 7.4, at both either resistant or non-resistant, our nanoreactor showed non-cytotoxicity, which also was in good agreement with this health and death essay. So when we try to understand why our nanoreactor can induce cytotoxicity to resistant cell, we first studied the PT, which is platinum, which is found in the cisplatin, internalization, cellular internalization, as well as the production of PT, or platinum DNA adduct. It was found that our nanoreactor can enhance the PT internalization into the cell compared to the free cisplatin. And also, there will be much production of PT DNA adduct. And also, it was found that nanoreactor can remarkably enhance the PT cellular internalization as PT DNA adduct in both resistant as well as non-resistant cells. Next, because our nanoreactor can produce high amount of ROS, it was designed to produce ROS. So we started the ROS production of our nanoreactor in the cell. It was found that our nanoreactor can produce high amount of ROS, which was also confirmed by flow cytometric experiment. And because the high production of ROS it is important that can reduce the various accumulation of pro-apoptotic pathways such as caspase-3. So we started the production of caspase-3 by using our nanoreactor in the resistant as well as the non-resistant cell. It was found that our nanoreactor can enhance the production of this caspase-3 in non-reactant as non-reactant in comparable manner, which was much higher than other formulation. And also high amount of apoptotic cell, which means the cell which die or will die later, was much higher compared to other formulation. So next, we test our nanoreactor in vivo, and then start by studying the bioavailability as well as intratumor ROS production. It was found that even after 24 hours, our nanoreactor can be accumulated at the tumor site. And also it can produce high amount of ROS at the tumor site. And also about the anti-tumor efficacy of our design nanoreactor, it was found that our nanoreactor can severely inhibit the tumor growth, which in a manner which was much higher or much efficacious compared to other treatments which have been used. And then our nanoreactor was somehow compatible, which means there was no cytotoxicity. So in summary, we have developed a nanoreactor that can improve the platinum uptake and show potential to inhibit drug resistance in lung cancer such as A549 and also inhibit the tumor growth in the vivo. Also, the nanoreactor can sensitize cisplatin against cisplatin resistant cell, mainly through the amplification of ROS reactive oxygen species and activation of pro-apoptotic pathways such as caspase-3 and also apoptosis. So in the part 4 of my talk is about the targeting mitochondria metabolic activity to overcome drug resistance. Here we are going to talk about the role of mitochondria organelle in the development of drug resistance in cancer cells. It is known that mitochondria plays a critical role in the cell, such as the energy source. And also, it has been reported that mitochondria organelle can also mediate several survival mechanisms in the cancer cell, even during the treatment or in the presence of various stress in the cell. And here we hypothesize that by development a dual drug targeting polymer prodrug nanoparticle, which can target mitochondria metabolic activity to reverse multi-drug resistance. So this is how our first we start by synthesizing a polymer prodrug, which is having a dual targeting, which can target the cell by folic acid, or which can target mitochondria by TPP, tyrosine phosphate. This polymer prodrug was containing three parts. This hydrophilic, which used the OHMA, and then this hydrophobic part was containing the cinnamaldehyde. This cinnamaldehyde, it is known to interact with the mitochondria and produce increased amount of ROS. And then this drug of our own, this is a common drug which is doxorubicin, and it can kill the cell, especially through the DNA damage and other mechanisms. And then like this, our dox, it can be released when there is high production of ROS. This monomer was responsive to ROS. For example, when our nanoparticle is injected, it can target only several cancer cells through the folic acid. And when reaching the cell site, such as it can release the ROS, it can release this cinnamaldehyde at lower pH, such as pH 6.5. And this cinnamaldehyde can interact with the mitochondria. And then within the release of DOX inside the mitochondria, it can disrupt the mitochondrial metabolic activity, which can easily kill the cell, even if the cell can be resistant or non-resistant. So we started by synthesizing a series of polymer. This polymer, they are only different from, such as this containing both cinnamaldehyde and DOX. And this one is only, DOX is missing, and this one is cinnamaldehyde is missing. This one is the same, but it has no targeting moiety. For example, as our polymer of interest, which is the first one, which has been abbreviated as PCD, PCD polymer, which has also been used to synthesize PCD nanoparticle. First, this is how it has been synthesized. It has been synthesized based on the rough polymerization, and then it was containing azide moiety, terminoazide, which can be used to conjugate either TPP or FA, folic acid. Next, we use our azide terminopolymer to conjugate TPP or folic acid to get the dual targeting polymer prodrug. And then after analysis confirmed the successful conjugation of TPP or FA to PCD polymer prodrug according before and after the quick reaction, which was copper catalyzed quick reaction that was used for this to conjugate either FA or TPP. We use our synthesized polymer prodrug to self-assembly into micelle nanoparticle. Then our nanoparticles show relatively low critical micelle concentration, which also it was found that our nanoparticles were stable at different conditions, either at different physiological conditions. For example, at PBS and also in the PDI, in the RPMI media, which is cell culture media. When we come back on our polymer prodrug, such as if there is low acid, such as pH 6.5, it can induce the release of this cinnamaldehyde. And then when this cinnamaldehyde is induced solely in the cell, it can interact with mitochondria and produce ROS. The produced ROS can come back and also help to enhance the release of DOX in the mitochondria. And then we started the DOX release at different conditions, and it was found that in the simulated tumor environment ROS, PCD nanoparticle can increasingly, such as according to time, release DOX at both 6.5 and also at 5.4 pH. We applied our prepared nanopolymer prodrug to study the cytotoxicity effect in the cell by using MCF7 cell ADR, which means these cells are resistant to dose. These MCF7 these cells are non-resistant. For resistant cells, there was much difference between either the treatment efficacy between free dose or our nanoparticle. This NPCD, which means this nanoparticle has no mitochondrial target or cellular target. It is the same as this one, the only difference is not having the targeting moiety. And also, it was found our nanoparticle were much cytotoxic to non-resistant cells in a comparable manner, even comparable to dose cytotoxicity, but in the resistant cell, the situation was not the same. Our nanoparticle showed much cytotoxicity compared to free dose, which was relatively non-cytotoxic to the resistant cell. So, in order to try to understand the mitochondrial trafficking competency of our prepared nanoparticle, we used this PCD nanoparticle, PD nanoparticle, and non-targeting PCD nanoparticle, as well as free dose. As can be seen, we use this blue channel to stain the cell nucleus, and then we use the green channel to stain the mitochondria. This is the natural fluorescence of dots. When we made it together, it has been found the dots can only be found at the nucleus side. Then this nanoparticle, which cannot target any organelle in the cell, it was found there was a co-localization, either the green and red were separate. However, these two nanoparticles, which are this one, which can target both cells, both cancer cell and mitochondria, the localization ratio was very high. It can be also confirmed by this yellowish color compared to other NPCD or dots. And then it has been found that PCD and PD nanoparticles showed high comparable effectiveness to target mitochondria. We also start the intercellular ROS generation because when there is independence of cinnamaldehyde, can use high production of ROS in the cell. It was found that our nanoparticle can use the generation of increased amounts of ROS within the cell, either through the resistant or non-resistant cell. It was exactly the same. We also studied the antitumor efficacy of polymer product. When it was found, it can also inhibit the tumor growth by tumor growth of resistant cell. And also it was the body weight, there was no bigger change by the free dose, which is commonly known with high cytotoxicity to the body. Here also we found that our nanoparticle can severely damage the tumor tissue. And also it is commonly known that dots is much cytotoxic to the heart, which was also found by H&E staining, but our nanoparticle showed no obvious change, no cytotoxicity to the heart. So in summary, we developed a dual targeting polymer product which can be used to prepare a PCD nanoparticle. Then our nanoparticle can target the cell and also the mitochondria, and can also amplify the ROS level and can release dots, and then can kill cells through the amplification of oxidative stress and also the damage of the DNA damage. And then this cancer treatment strategy showed potential to reverse MDR in cancer treatment and achieve enhanced therapeutic effect both in vivo as well as in vitro. So I come to the conclusion. It can be suggested that the application of therapeutic nanoreactor to treat cancer suggested a promising approach to achieve versatile treatment strategy. And also the targeting mitochondria metabolic activity showed also a promising strategy to effectively treat tumor and reverse drug resistance, which is often encountered in cancer. Finally, our work presented in this thesis is expected to inspire the future development of advanced therapeutic nanoreactors as well as the development of more advanced therapeutic strategy which can also reverse the drug resistance in cancer treatment. Here is the list of applications, and before I end my presentation, I want to thank my supervisor, Professor Gar. I'm also grateful to all my lab mates and classmates. They were much helpful during all this work. I'm also thankful to the USTC as well as to us for the financial support, as well as my university, which allowed me to come here for my studies. And also, this is the end. Thank you for your kind attention, questions, suggestions. You are welcome. Thank you.
There is no interesting.
Shall I start? Okay. One tiny question. I noticed that you always use essay, the word essay, afterlife and dead essay. Essay is a type of, how to say that, a type of article, a type of what you wrote in English. You should change that to essay, A-S-S-A-Y, rather than E-S-S-A-Y. Okay, back to your first project. Your first project, the first, I think the second section, which corresponds to. You mentioned that you want to.
This one?
Yeah, this project, but go to the in vitro cytotoxicity slide. Yes. This one, this slide. I noticed that you performed the IC at pH 6.8. In your introduction, you mentioned that the goal of this project is to upregulate the intracellular ROS level. But pH 6.8 is the pH of the extracellular fluid within the tumor tissue. If you want to upregulate the intracellular ROS, you may want to think about where do your nanoparticles stay after they are internalized by the cells. Maybe most of the time they may stay within the endosome. And within the endosome, the pH is much lower than this pH. I'm not suggesting that the cytotoxicity will be less than what you see at this pH, but it may be even stronger. But from the perspective of logic, you may want to use a pH of like 5.5, something like that.
Okay, thank you for your suggestion.
Yeah. But this is okay, but my point is that this pH corresponds to the extracellular pH, and to demonstrate that your nanoparticles performed what you expect them to do, you may want to use much lower pH, which corresponds to the endosome pH. Okay. Sure. That's where I'm here for, right? The third project, the one you reversed the multi-drug resistance by release dots. I'm very curious.
Yeah.
I'm very curious about the target of dots.
Target of dots.
Yeah, free dots.
You mean the type of project, right?
Yeah, the fourth section. Yeah. What's the target of free dots? Because your nanoparticle functions by releasing dots.
Yes.
In other words, its cytotoxicity comes from the cytotoxicity of free dots, right?
Mm-hmm.
So what's the target of free dots?
The target of free dots.
Yes.
For example, here we use free dots. And
Yeah, I understand you use free dots to show that your nanoparticle can kill both resistant and sensitive cancer cells, but the free dogs cannot. It shows that the resistant cells are really resistant.
Okay. So
My question is that the reason why your nanoparticle can kill dogs resistant cells may arise because because the higher internalization efficiency of your nanoparticles as compared to free dogs. Because the resistance, I noticed that you did not really use any specific, any characteristics pathway within the resistant cells. So the better performance of your nanoparticle compared to free dogs becomes from the higher internalization efficiency of your nanoparticles, likely because they can enter the cells through endocytosis.
yeah, actually, as you know, mitochondria also is having its own DNA. And then when we use our nanoparticle, what you say, it may be true. High encapsulation or high internalization of dogs can also enhance the cytoplasmic system. But also our nanoparticle has that we are much targeting the mitochondria. It means when the dose is released in the mitochondria, it can disrupt or it can damage the mitochondrial DNA. And also in the presence of high amount of ROS, the mitochondrial metabolic activity, which can be also disrupted, which many papers also suggest that mitochondria contribute a lot into the generation of resistance in the cancer cell.
Did you use, go back to your design of this nanoparticle? The design. You mentioned that it's a two-step release mechanism. First, the proton cut something from your polymer, and that something react with something else to produce peroxide, and the peroxide then function to release dogs, right?
Yes.
Okay. So, did you use any nanoparticles that cannot go through this mechanism as a control to see if you simply encapsulate dogs into nanoparticle, can you still see enhanced cytotoxicity to resistant cells?
Okay, that's good suggestion, and I think in the remaining experiment about this project, I may include the cytotoxicity by taking another nanoparticle which can encapsulate such as dogs.
Okay, your work is already very good. I'm just think about from simple perspective of logic.
Okay,
I'm trying to understand what you are trying to prove. Okay, your work is already very nice.
Thank you so much.
Thank you.
I guess I have only one question. So you said on your paper, quote unquote, cinnamaldehyde inhibits, I guess, the MDR action through in situ amplification of mitochondria, ROS, right?
Yes.
So I guess my question is how could you say for sure it's this particular pathway you're talking about? You don't have a control that is, for example, benzaldehyde. How could you know it's not simply the action of the aldehyde group that's damaging your cell and not this particular pathway?
So what you say is much understanding of the logic. So about this experiment, I think in order to be more sure that the cell are dying because of mitochondria.
Yeah, you were saying it's possibly the release of cytochrome C that's damaging the cell, but aldehyde itself is toxic, right?
Okay.
So how could you know it's not simply the aldehyde that's reacting with something, I don't know, on the mitochondria?
Actually, this work is still under process, and in the remaining experiment which I want to perform, so I want to study the release of cytochrome C, which can also have which can suggest whether it is about mitochondria or only the ROS is killing the cell. I think that's a good point. So I will focus on
Right.
the
So
experiment.
I think it's less convincing if you're not showing, let's say, a control experiment with benzaldehyde.
What he is suggesting is that you need to add another set of
Right.
thermal control.
Mhm.
Yes, yes.
Yeah.
It's almost quite like what he suggests to me. Anyway.
Could you please go to slide 28? I think the NL spectrum of the polymer is wrong.
Here,
You can't find benzene. Where is the benzene?
here, because I mean, maybe the benzene is about more than 8. I need to check. I
No,
have
no,
the-
no, definitely not. You should check the NMR spectrum. I
Okay,
think
I will
this
check
one is
again.
wrong. And also, could you please go to slide 45? Yeah, so why are there two dots channel? For example, you can find the left one is dots and also the top panel is all dots. What does that mean?
You mean the left one? Like,
Yeah. So you label
Oh,
them.
here,
Yeah. Why are there two dots?
here.
Yeah.
This means I use 3 dots for treatment. This is the treatment group, and which also we having the cellular nucleus, mitochondria, and dots. And also this is, for example, this is nanoparticle. So there will be nanoparticle also containing dots. This means there will be fluorescence.
Oh, I mean, if you overlay the blue channel and the red channel, what does that mean?
Blue channel and red channel.
Yeah.
What does it mean?
Yeah.
This means if we overlay, it means dots will be, we can decide whether dots is in the nucleus, cell nucleus, or is in the cytoplasm.
No, I still can't understand why are there two dots channel?
2 dots channel.
Yeah.
Like this one and this one.
Yeah.
This is, maybe I should change this one. This is the treatment group which we use 3 dots.
Okay.
3 dots.
Yeah.
And this is the dots channel. This is not channel. The channel is this one. This is the treatment group, which
Okay.
has been treated with 3 dots. Maybe I will use 3 dots to avoid the confusion.
Okay. And another problem, I think you use the TBP for the mitochondrial targeting, right?
Yes.
But I found the chemical structures in your, for example, in here,
Yeah.
the spacer, I think you have 4 carbon. But in your thesis, I found you show the chemical structure of the TBP products. They only use the 2 carbon. I think the chemical structure is wrong.
Because first we modify the TBP with alkyne, the alkyne,
Yeah.
such as it can react with the azide polymer. So if there is wrong about the number of carbon which I put here, I will go and check and try to write what is correct.
So could you have your thesis?
Yes, I have.
Yes, could you go to 144?
1.4.
Yes. So you use this one for the TDP for labeling, right?
Yes.
So chemical structure is definitely wrong.
Okay. So I will go and check again and write the correct structure.
Yes.
Thank you so much.
Yeah, you did very well. You have very good packages.
Thank you.
Okay, I have another question. So you have synthesized different large carriers for the cancer therapy, and all of them have very good therapeutic effect. So, for example, if you want to use your large particle to treat one kind of cancer, how do you choose? Which one is the best?
Actually, you can choose according to different factors.
Just for example, lung cancer or liver cancer, some kind of cancer in your body. So which one is the best?
I understand. Like, this is a different nanoparticle, which can also work in different environments. Such one can be more specific to either the cancer cells which are resistant or non-resistant. The other cannot be effective only to non-resistant cell. Actually, if you want to choose, maybe I think because if you want to choose, you can choose this one because it is easier to self-assembly. But when you come back to the polymorphism, it's not easy to control. Today you can get good one, tomorrow no. So I think the mycel one are the ones which are easy to make and which you can reproduce.
Thank you.
Okay, hi Felix. Good job with the nanoreactors to deal with the drug and to reverse the drug resistance. So I have questions. So jump to the slide 27. So how did you load the cisfatty inside of the nanoreactor?
First,
because the cisfatty is hydrophilic drug.
Yes. Because nanoreactor is having the inner cavity is hydroaqueous solution. The polymorphism can encapsulate both hydrophilic and hydrophobic. The hydrophilic can be encapsulated in the inner aqueous solution inside. And then hydrophobic can be embedded, for example, in the membrane. So about how we encapsulated the cisplatin, first we dissolved cisplatin, we took 5 milligram and dissolved into 5 milliliter. And then that we dissolved our polymer in the THF, and then our cisplatin was removed, and then we did self-assembly by injecting. So, and then after the self-assembly, we removed the unencapsulated cisplatin by dialysis. So that's how we was able to encapsulate the cisplatin in the aqueous solution of the polymer.
And so the cisplatin will, during the drug release process, the cisplatin will release the chloride. So, and then the platinum will conjugate to the nitrogen of your compound. You see that? It's very easy to happen, this reaction. The nitrogen will conjugate to the platinum. It's tricky. So do you know the mechanism of the cisplatin reacted with the DNA? The cisplatin will release the chloride and then distort the DNA and then so it kills the cancer cells. So about the reaction between the nitrogen and the platinum.
Thank you for that concern. Actually, when I was doing this, I didn't think about that, but now as I know, I will go to search, and also if I can put any recommendation about the further research, which will be quite similar to mine. And if that reaction can happen, because
that's the tricky part.
Yes, I understand. I will go and find about that reaction, and also I can do some recommendation in my thesis about further experiment.
Okay, go up to the slide 23. So, on the tumor growth curve. So I saw that the red line, the tumor stopped growing or there's no tumor. So in some medical school, lots of many supervisors want to see the tumor growth curve go up, not just to stop there, stop on that day.
Oh.
So I would say, is there tumor on that mice? I don't know.
Yes, because here there is 0 and here there is around 100. So
Did you any section of that tumor, of the red tumor?
Here is this one.
It's seen highly, right? So now on the
When you remove
on the red line, there is no tumor, right?
when you remove the skin, the tumor becomes tiny. But when it's still inside, you can touch, and you can even measure the size.
So I wonder if your drug kill the tumor or there's no tumor at the starting point.
There is still tumor at the stop point, but the size is smaller compared to other treatment.
Okay. Very relaxed. I just ask you some questions. What do you think of the future of the drug delivery?
Actually, it's quite complicated question because whatever we are doing.
Because you know why I ask you this question, because the NCI just shut down the department, the project of the CCE which supports the nanodrug for the tumor therapeutic.
You know, when we do this, we all rely on the EPR effect, and
not only EPR effect.
As you know, many scientific researchers say EPR are only effective in small animals such as mice, but when it comes to humans, it is not effective. So about the future of nanotechnology, maybe it's better to think more and think how to change, how to improve the application of nanotechnology, and especially with the tumor accumulation of nanoparticles.
Thank you. Okay.
Only one question I think is what kind of polymer you choose
For example,
you understand my question?
yes, I understand what kind of polymer of my choice. Yeah. For example, this polymer, it is easier to make and easier to control the DP, but also making polymers is not very easy. So if we say the polymer of my choice, I can choose to make this one, which is easier to make. But when you come to this polymer, for example, this one, this polymer, when you prepare this doxymonomer is not easy for polymerization, and it's not easy to control the DP. Today you can do the same condition, but not the same DP. Tomorrow, not the same DP. So this one is quite. Also, it can take long time to make.
Okay.
So my choice would be the first one.
Say again. Okay.
Thank you.
这里的各位老师、亲爱的同学们，大家上午好，我是高中期17级的博士生柯文东。今天很荣幸能够在这里向大家汇报我这几年来的工作。然后我的